I am Mitchell Kronenberg. I'm a longtime professor at the La Jolla Institute. I was president for 18 years, chief scientific officer. I'm currently professor and president emeritus. Mate cells are a very special population of T lymphocytes, and they act differently from most other T lymphocytes. One aspect of that is most T cells see peptides, and these cells see riboflavin metabolites. Another aspect is they tend to go to tissues, particularly the liver and lung. And a third aspect is they make very, very rapid responses. And a fourth one I should mention, if it's not overwhelming, is they have the same specificity across individuals. And that is really the key, because most of our T lymphocytes are individual or nearly individual in the sense that what they recognize is based upon our tissue type. And our tissue types vary greatly. For mate cells, they do not vary between person and person. In fact, they hardly vary between mice and humans. So over 60 million years of evolution, mate cells have really not changed their specificity. So that is really a surprising fact. The thymus is the organ where T cells go to school, in other words, where they differentiate. And it's not easy to get human thymus. But in children who require cardiac surgery, they remove half the thymus in order to get it out of the way so they can work on the heart. And they pretty much aren't using the material. That we connected with the surgeons, and they thankfully, they provided us with this material so we could analyze the mate cells where they're born in the human thymus, as well as in the lung and in the peripheral blood. Those are the three human sites that we looked at. We've been interested in these T cells that have very conserved specificities. There are other populations that have a similar property. And one reason is to understand how they function, how they evolved. They're so different from what you might call the mainstream of T lymphocytes. But the other thing is, because they're the same across individuals, they can easily or more easily be used as cell therapies, in which my mate cells could, in principle, be given to you, and they won't cause graft rejection. While my CD4 or CD8 T cells, my main population of T cells, would cause graft rejection. So there's both a fundamental curiosity aspect to research on these cells, but also the possibility that they could be used therapeutically.
I'm Gabriel Ascui. I'm in the Mitch Kronenberg lab here at LJI. I'm also part of the biomedical science program at UCSD. I think the most important thing is that we're looking at cells that have the potential to be used broadly and therapeutically. And I think the more that we understand these cells that have the possibility of not being restricted to single individuals, but can actually be transferred to other people, the more we can think about new therapies that could involve these type of cells. So despite that, in this particular study, we were able to obtain a lot of human tissue from different places. It's still important to look at mice because we can get a more systemic view of what's happening. Mate cells are cells that you can normally find inside of tissue. They're not circulating like most T cells. So we wanted to look at a lot of different tissues and see differences that we can find in their function and what they're doing, and potentially if they could be transformed from one type to another. These particular cells, they're called innate-like T cells. And we study not only mate cells, but other types in this lab. And in general, what I would say is that they seem to have this innate-like characteristic, which basically means that they act very quickly. So they're kind of like your first line of defense. That's why they like to be there where there's more exposure. And particular mate cells, they like to be in mucosal tissues, so tissues that are exposed to the environment, like the lung, like the gut. So we used a lot of single-cell RNA-seq. We wanted to look at transcriptomical differences that we could find within these cells, how their gene expression was working. But we wanted to look at a level that we could see a single cell and distinguish, well, what happens if the cell is inside, let's say, the liver versus what happens if it's inside the lung. But additional to that, maybe there's different types of cells within that same tissue. Maybe there's different types of cell within the lung. So to actually be able to look at that and to graph a representative population, we use this technique that allows us to have higher resolution of what we're looking at. So if we could make normal cells more innate, this means that these cells would be quicker, they would respond, let's say, more vigorously, and that would allow them to, for example, arrive to tissue quicker and being able to combat any type of infection or cancer or other type of disease more quicker. So this is one avenue that we're looking into, like what makes a T cell innate.